Hey guys, I just wanted to quickly go through the notes that I posted on Google Classroom for you earlier today. So, we're going to first talk a little bit about cells, and the reason why we're doing this is because our next lab actually looks at what happens at the cellular level when the blood glucose changes. So, what happens to cells in the bloodstream when blood glucose is too high and too low? And this all deals with cell membranes. And cell membranes actually surround all cells, whether they are prokaryotic or eukaryotic. We're going to focus on eukaryotic cells because those are the cells that make up the human body. Now, just as your body needs to maintain homeostasis, your cells also have to maintain homeostasis. And one of the ways that they do that is that they control the movement of substances across their cell membranes. They are selectively permeable, meaning that some things can easily move in and out of the cell, whereas other things have to work a little bit harder, sometimes using energy. Now, if it uses energy, it's called active transport, and it uses ATP that's made in the mitochondria of the cell. Now, we're not going to really talk about active transport in this class at this time. However, you will be talking about this, or you have already talked about it in biology. So we're going to be talking about solutions a lot with this activity, and I wanted to review what a solution is and what the different parts of solutions are. So a solution is a type of mixture, it's a homogeneous mixture, and that just means that it's a mixture in which the molecules of different substances are thoroughly combined. It's made up of a solute and a solvent. The solute is the substance that is dissolved in the solution, and the solvent is the liquid that does the dissolving. So if we're talking about blood, the solute would be the glucose and the solvent would be the blood plasma. Another example would be Kool-Aid. In Kool-Aid, the solute would be the actual Kool-Aid powder and the sugar, and the solvent would be water. Now, cells actually live in solution. That would be their environment. If they're blood cells, they're surrounded by plasma. If they're in an organ, they're going to be surrounded by water in that organ. But they also have solution inside of them, which is the cytoplasm. Now, here's some functions of the cell membrane. The first one is going to be the one that we're most interested in for this activity. And what the cell membrane does is that it regulates passage of material in and out of the cell. It also is going to provide limited support and protection to the cell, and it houses proteins that allow the cell to communicate with other cells. So let's go ahead and take a detailed look at what the cell membrane looks like. It's made up of phospholipids, and those phospholipids have two parts. They have the phosphorus head that is hydrophilic. This is polar, meaning that it likes water, water, philic, to love. And then we have the lipid tail that's hydrophobic. Remember, lipids are very polar. These are like water hating, and these hydrophobic tails are what's going to keep out most of those substances that will pass through protein channels or through active transport. We're going to talk about that in a couple of slides. So the cell membrane is also called a phospholipid bilayer. The reason why it's called that is because it has that double layer of phospholipids. Now, looking a little bit closer at this picture, there are other structures that are located on the cell. And we have the peripheral proteins. This is the green olive looking thing that has that weird tree thing sticking out of it. That weird tree thing is a carbohydrate antenna. And what these cells do, or what these structures do rather, is they're going to pick up messages from outside the cell from other cells and relay those messages to the nucleus. You also have cholesterols. The cholesterols are going to be located actually in between the phospholipids. They prevent the cell from freezing. And then you have protein channels, which are going to allow large molecules to pass through them. Another word that's used to describe the cell membrane is fluid mosaic. And the reason why we use this term is because it's flexible and not rigid. Another property, like we've mentioned before, is that it's semi-permeable. Another way of saying that is selectively permeable. And again, this just means that some materials can easily pass through the membrane, whereas other materials have a harder time and will need a little bit of help. So, There are different types of transport. We're going to be focusing in this class mostly on passive transport. As I said before, there's also active transport. Passive transport just refers to any movement across the membrane that does not require energy expenditure. It does not require ATP. 3 types of passive transport are diffusion, facilitated diffusion, and osmosis, and we're going to talk about those more on the following slides. All right, so first up we have diffusion. So diffusion is just the movement of molecules from a high concentration to a low concentration. This just happens naturally. Think about someone that is peeling an orange across a classroom. When they're peeling that orange, you can visually see them peeling it, but you might not smell the orange oil yet. However, the people that are right by them will pretty immediately smell the orange oil. Eventually, the orange oil will diffuse from that area of high concentration, where the person's peeling it, to the area of low concentration, where you are sitting. That is diffusion. Here are some more examples, and these are things that are going to diffuse across membranes. Molecules that easily diffuse are going to be small molecules and also nonpolar molecules. So oxygen and carbon dioxide, those are the 2 molecules that are pictured. They're both small and nonpolar. They will easily diffuse right across the cell membrane. Other molecules will have to use facilitated diffusion. Now, facilitated diffusion is just the diffusion of large or polar molecules with the help of proteins. So here are some examples of molecules that will need to move across the membrane through facilitated diffusion. They are glucose, if we're looking there on the left, and water there on the right. Glucose is too large to just go across the membrane, and water is too polar. We're going to talk more about the diffusion of water in just a couple of slides. There's also diffusion through ion channels. Ion channels are just a special type of transport protein that allows specifically ions to go through. So it's a transport protein that has a polar pore that allows those superpolar ions that again would be blocked by those hydrophobic tails, those nonpolar tails, and the ion channels will let them easily pass through. All right, so osmosis. Osmosis is the diffusion of water across a membrane, and the reason why we're talking about this is this is what our lab is going to focus on tomorrow. So when solutes can't follow the concentration gradients, typically water will respond. So if we're looking at the case of diabetes, where glucose can't pass across the cell membrane because of either insulin resistance, or in type 1 diabetes, there's no insulin to allow the glucose to go through, water will respond and try to maintain that equilibrium. Basically, by changing the water concentrations inside and outside the cell, we can reach equilibrium without moving the glucose. And these photos that are on this slide just show typically what would happen in unequal situations where water would have to move and the solute was not moving. So if we have 2 cells, I'm looking at the bottom left-hand picture with those 2 blue blobs and the red and white dots on the inside. In the cell on the left of that picture, we have a much smaller amount of solute. Those are the red dots. On the right side of the picture, we have a lot of solute. So what will end up happening naturally is that the water will move from the area where the water is at high concentration to the area where the water is at a low concentration. So water is going to flow from that left cell into that right cell to try to maintain equilibrium, to try to basically dilute the solute in that right cell. Now on the right-hand side of this slide, there are 2 U-tubes, not the app. They are tubes that are divided by a semi-permeable membrane. Ask me in class if you want me to explain what's happening here. We're really not going to talk about U-tubes in our class, but you will talk about those in biology. Really what I want to focus on are osmotic solutions. So there are 3 types of solutions. We have hypertonic solutions, isotonic solutions, and hypotonic solutions. In a hypertonic solution, the water concentration inside the cell is higher than outside the cell. You also might hear a hypertonic solution referred to in terms of the solute. So in a hypertonic solution, the solute concentration is going to be higher outside of the cell than inside of the cell. In an isotonic solution, both the water and solute concentrations in a cell are equal to the water and the solute concentrations outside of the cell. Now, in a hypotonic solution, you have the water concentration inside the cell. It's actually lower than the water concentration outside the cell. And if we're looking at it in terms of the solute concentration outside of the cell is going to be lower than the solute concentration inside of the cell. So let's look at what happens to cells in these different types of solutions. So like I said before, in a hypertonic cell, we have more water inside the cell than outside. Water behaves just like any other material in diffusion, and it's going to move from the area of high concentration to an area of low concentration. So water is going to move out of the cell, and the cell will shrivel. In an isotonic solution, there's no net movement of water because the concentrations are the same on both sides of the membrane. This means that animal and plant cells are normal, or another word for this is that they are flaccid. Now, in hypotonic solutions, we have water moving inside the cell. In this case, we have more solute inside the cell than outside the cell, so water moves in to try to maintain that equilibrium. And if too much water moves in, animal cell membranes can actually burst, but plant cell membranes will not burst because they have the cell walls that will prevent that from happening. Now we have our last slide here. This is kind of a question for you, so this is like a review. If we have red blood cells that are inside the cell have 0.8% sodium chloride or salt in the cytoplasm, and we place those in a solution that contains 1.5% NaCl, what will happen to the cells? So if we have more salt outside the cell than inside, where will the water move? If we have more salt outside the cell, what's going to happen is there's going to be more water inside the cell. That water is going to move from an area of high concentration to low concentration, meaning the water will move outside of the cell and the cells will shrivel. All right, guys, that's going to wrap up our notes here. Please email me if you have any questions about these, and we will talk about these very briefly tomorrow. Come in class ready to talk about this and also ready to do your lab. Thanks, guys, and I'll see you tomorrow.